Hello, everyone, and welcome to our Friday Rare Round session. I do want to encourage everyone here, if you're comfortable, to please turn on your video camera. We would love to see your faces and just have a more intimate discussion this time around. So if you're comfortable, please don't hesitate to turn on your videos. Oh.
Left.
No worries. We'll be waiting for a few more people to join, and then we will get started. Then welcome for those of you who just joined us. Please feel free to turn on your video camera if you would like. We would love to have a more intimate session this time around. So please feel free to turn on your cameras. We'd love to see your faces. Welcome. We're going to get started in just a few. We're going to wait for some more people to join. How's everyone's day going? You want to write in the chat box where you guys are coming in from, if you enjoyed our opening session today.
Yeah, I hope everybody's doing well. And is having good weather.
Yes.
I think in both coasts today, because it looks like your weather is great, and up here in Philadelphia is beautiful too. So
yeah, I mean, this is pretty constant. It's like,
yeah, you guys are in the lucky spot.
Yeah.
Yeah, we are pretty spoiled over here with the weather.
Yeah. No, we're having a good day on this side of the country.
Oh, that's great.
Sunny,
Yeah.
and I think it's a little bit too hot for us, but that's what it is.
Yeah, this is my favorite time of year in the East Coast, when right before it gets too hot and too humid.
Yeah, I think it just rained so much in here yesterday that and it's hot, it's sunny today, and the humidity is huge. It's really bad.
Yeah.
All right, so we're just gonna wait one more minute, let some more people join on in right now. There we go. And for those of you who just joined us, welcome to the Rare Round session for cell models and rare disease research. We would love to see your faces if you feel comfortable with putting your webcam on. If not, totally fine as well, but we would love to see who you all are. All right.
I completely understand the camera thing. Like it takes, yeah. I'm so not used to going to work anymore that it takes me so long to actually get ready to put my face on camera that I completely understand.
Yeah.
It's like a completely different thing nowadays.
All right, so let's go ahead and get started. Thank you so much to everyone joining us here today for our Rare Round session on cell models in rare disease research. Before we kick things off, I do want to start with a few brief housekeeping items. So currently, right now, you are all muted to reduce any interruptions, but throughout this chat, if you would like to unmute yourself and ask a question, we do encourage it, or if you would like to place your questions in the chat box, you may do so as well. We just do ask to please keep yourself muted when you aren't speaking or when someone else is speaking, to not cause any distractions. So other than that, we are going to get started. I would like to introduce our two discussion leaders here today. First, we have Deborah Requesens from the Coriell Institute. And we also have here today Herve Tiriac from the University of California, San Diego. So I'm going to turn it over to them real quick to give a brief introduction of themselves. So we'll start with you, Deborah.
Hi, hello, good afternoon and good morning for those of you on the other side, on the West Coast. So my name is Deborah Requesens. I work for the Coriell Institute for Medical Research. We are in Camden, New Jersey, just outside Philadelphia. We're a nonprofit that is well known for being a biobank for many decades now. I am the principal investigator of one of those biobanks, the NIGMS repository. We are a repository of cell lines and DNA that specializes in rare diseases and chromosomal abnormalities.
Great, thank you so much.
And hello, everybody. I'm Herve. I work at the University of California, San Diego, where I basically study pancreatic cancer mostly, as well as other forms of cancer. And to study this, I, along with other scientists, are generating patient-derived cellular models, which we call organoids, which in a dish recapitulate the tumor of the patient from which they're derived. And we use those for precision medicine research to see if we can predict the outcome to treatment in a dish before the patient is actually exposed to the drugs. So that's what we do. And I have a bit of experience with different cellular models throughout my research.
Thank you. And then at this time, I would love to open it up to any of our attendees here. If you would love to unmute yourself and introduce yourselves to the leaders, maybe tell your name and what brought you here to this discussion. At this time, if you would like to unmute yourself, the mute button is at that bottom left-hand corner. Please feel free to do so. So I'll give you guys some time if you want to do that. We'd love to hear. Hi there. All right.
Hi, my name is Kim. I'm an assistant professor at the University of Alabama at Birmingham, and our group is trying to find small molecules to suppress termination at nonsense mutations as a way to treat a variety of genetic diseases. And of course, in doing this, we're very interested in utilizing different cell models. And I'm very interested to hear, more of the availability and perhaps some of the caveats in using cell models for, developing therapies.
Awesome. Thank you so much for introducing yourself, Kim. Anyone else would like to introduce themselves and what kind of brought them here to this discussion today? All right. No worries at all. We will go ahead and move along. So once again, if you have any questions, please feel free to place it in the chat box or feel free to unmute yourself and ask them once our speakers are done answering questions. So, all right, we're going to get started. So our first question we have, what are the, yes?
Maybe we can just, because the first question is basically what Kim just asked, the limitation of the various cellular models. Deborah, would you like to start on the availability of the models through the biobank, maybe?
So I guess so there are, of course, a large variety of cell models that you can use in research. I work on that first stage on the most basic 2D type of models, cell lines. We at Institute, we both work with both lymphoblastoid cell lines, which are immortalized cells, and that we make from blood. And we also make something that we call fibroblast cell lines that are made from skin punches. These are highly utilized in basic research. We offer tens of dozens of them, and we mainly use the LCLs being used to obtain DNA from them, which is also a big resource that we offer our repository. At this level of cell model, we are talking about cheap, fast, easy to obtain the samples from the patient, and easy to prepare the cell lines ourselves and to be distributed. So it's in a particular case of a repository sponsored by the NIH, these are free to the submitters, to the patients, and at cost for the investigators. And then after that, I guess you can get way more complex, and I think Herve has more experience on the more complex levels of models.
Yeah, so I've been working for the past, I don't know, now it's going to be 8 years or so on organoid models, which are three-dimensional models of both disease and normal epithelium. So we can derive these from gastrointestinal tract organs very easily, the GI. So the intestine works really well, the pancreas works really well, the liver also works really well. And I've been specializing in making these models from pancreatic cancer patients, as well as genetically engineered mouse models that recapitulate the disease. So pancreatic cancer mice, basically. Those are available in terms of can you obtain them and work with them in your laboratory. They're also easily derived from samples in your own laboratory, especially if you can get access to a mouse tumor. That's a very easy place to start. The patient tumors are a little bit more tricky because of all the regulatory requirements that are necessary to be able to obtain the tissue. But as long as the institution or your collaborators are able to provide just a little bit of the identified tumor piece from a patient, the methods are actually quite reliable and easy to implement with a little bit of an upfront investment, both in teaching and in materials to get yourself started. But it's not impossible to pick up. And in terms of commercial or other available source, the NIH is actually sponsoring an initiative called the Human Cancer Models Initiative, which the goal is to generate organoid and iPS and other types of cell lines for rare diseases and deposit them with the ATCCs so they can be provided pretty much at cost to researchers. There's going to be a bump in price for commercial entities, but for researchers, it's pretty much at cost. So there's definitely avenues to be able to obtain these models now or to make them if you wish.
It's a good thing that you mentioned the iPSC. So that's actually the newest investment of the NIH with us as well. We have started creating our bank of iPSCs from rare diseases, and we also offer at Coriell the possibility of making them if you wish and you don't wish to deposit it within the repository. So that's becoming more and more available for researchers. And so I just guess, because we did this yesterday, I guess we kind of know the questions, so I'm just gonna keep going the conversation throughout the questions that the Canoe asked yesterday. So we were asked yesterday about the cost of this, and Herve mentioned some, so the very limited cost of doing something as big as an organ. For a cell line, if you work with a repository that is sponsored by the NIH, they are usually free to anybody that would like to participate in research. And this is a great opportunity at this time where the rare disease patients and advocates are so involved in research. They can definitely participate by giving their samples. So in our case, it can either be blood or skin, and then, you know, to no cost. You can just go send our samples, and the cell lines will be established and distributed. And we do also have a tier pricing for commercial and academic, which is just a little bit different. But even in the case of commercial laboratories, it's still very much at cost since we are sponsored by the NIH. So on the other hand, laboratories such as ours and many other companies, you can get your iPSC established. For example, is, you know, around, I would say, $10,000 to $12,000 more or less. So that does take a little more of an investment to begin with. But there's a possibility of doing that as well.
Thank you. All right, so going off of that, what are some new advances in tools or technologies that can facilitate development of cell models?
Yeah, I can get started on this one. So there's a lot of new cool tools for researchers, as I'm sure you know. A lot of the single cell sequencing technology has come to the forefront in the last few years and is very popular right now. And it's been actually a real boon for the study of cellular models because we've been growing these models for a while, and now we can analyze the heterogeneity of the models themselves using the single cell technology. There's also a lot of metabolism type instruments to measure the flux, the production of ATP and respiration and things like that in the models with the seahorse. So there's a lot of cool things you could do in the laboratory now. And in particular, what I like about what I'm working on, specifically the organoids, is that you can make them from pretty much any patient with a pretty high success rate. We're averaging about 70% success rate. to establish these models from patients. So we can create larger cohorts of patients of models that we were not able to generate previously for the same cost and for the same time. So we can now do really cool studies where we have large cohorts of models that really represent the phenotypic heterogeneity that you see in the population.
And I think yeah, go ahead. No, I was just thinking that one of the coolest things that we have, and it has to do with cost, is how cheap it is now to characterize these cellular models. You know, as before, even doing a single sequence, and I don't know, you know, I feel really old when I think about you could just publish a PhD thesis when you sequence one gene. Back in the day, like now we can do this so easily. So our cell lines can be highly characterized. We can do all sorts of things for a very small amount of money. So I think it's a great advance in tools, and we can do not just, you know, the single cell sequencing, but we do a lot of epigenomics in our laboratory, for example. Yeah. Our company is moving towards cancer research a little bit, and that's a big part of it. So I think that's something that has been growing a lot, and that people are more interested to see what happens in that area. That is not just what we thought the DNA was DNA doing what it was doing. Now we've known for a long time, but now we can easily and cheaply understand the epigenomics of the models as well.
Great. And then, I'll just follow up. Sorry, just one second. No, one of the things we were talking about yesterday that Deborah mentioned that was really interesting was isogenic cell lines. So I'll let you continue on that because I think it's a really cool tool.
Well, we do a lot of this. So an isogenic cell line is simply a line that is exactly like the patient-derived line, but if it was a patient-derived line with a mutation that's causing a disease, we take that away, right? So it becomes a perfect control in which we have everything else in the background of the cell line, but it doesn't have the disease. So we've been doing this with, you probably all heard about CRISPR-Cas9 technology, so it's gene editing, and we are able to take out even chunks sometimes of DNA to replace where the disease was, the gene that was causing the disease. So these isogenic controls are now very used, and actually for most people doing research and people are trying to publish, they need isogenic control. So I think that this can only happen thanks to the technology of gene editing.
Thank you. All right, so next up, what are some key misunderstandings on this subject, and what are some common missteps by advocacy groups and by researchers?
I started last time. You want to start this time?
Yeah, I'm trying to remember what I mentioned yesterday because I thought it was good.
Okay, I'll start with mine, and then you remember what you said.
Yeah.
So one of the main things is just to, and again, the preamble to this conversation is not, it doesn't mean like it's misinformation or missteps. It just sometimes it's just lack of information, right? So many people come to me and have said, oh, I gave you, this, you made me is this an iPSC when we're talking about an LCL? So a cell line doesn't necessarily mean one thing or another. So there's a big difference between just creating an immortalized cell line compared to what we're making an induced pluripotent stem cell line. So those are kind of misinformation that I constantly have to explain people. Timelines, people think there's something that is fast. That you can, what's happened to my line? I gave you blood last week or 2 weeks ago. So these are living things that take sometimes. They take time, and sometimes, and I think it's a question coming out later, but sometimes they just don't grow. Sometimes they just, we baby them, like we say in the lab, and we do as much as we can, but sometimes some cell lines will not grow. They will fail without us understanding. So those little things can cause problems in the communication with researchers and with advocates.
Yeah, and I do remember now. So yesterday I mentioned something that is dear to my heart. So when we publish a lot of these really cool studies with patient-derived models, and we demonstrate that in the research laboratory, some of our models can predict the outcome of chemotherapy, for example, for a pancreatic cancer patient. A lot of time those results get disseminated in the news media through the PR of the institution, through the PR of the foundations that support us. And it's good for our results to be out there. But it does cause a little bit of a misunderstanding about where we are in the clinical utilization of the models. So we've been a few times surprised by having patients or patients' families call us at the laboratory asking to see if they can donate tissue so we can replicate the same experiments that we've published on their own samples. And it puts us a little bit into a difficult position because typically the tissue arrives to us in a de-identified for research purposes manner. Through very specific IRBs and very specific consent forms that the patient signed before the tissue is donated and de-identified. So it's a two-pronged issue. The first one, it's probably a communication issue from our end. We should be very clear that this is still research and not clinically implemented, and these news stories have to clearly label that, that fact. And on the other end, it does create this issue where there is a need for patients and patients' families to be able to participate in the research more actively. But there's few paths right now because of the IRBs being so protective of patient information for them to directly contribute to the research and directly be involved in the research, since we're supposed to be, not communicating with them. So that touches on another question that we're going to talk about later, I think, the regulatory hurdles and what patients can do about it. But it is important that patients that want to be involved voice that the need for them to be involved, so that institutions can listen and create IRBs that and consent forms that allow them to be more involved in the research.
Actually, that brings me to a thought that I had yesterday. I don't know if I mentioned it or not, but one of the confusions that needs to and causes a lot of misunderstanding between researchers and patients, it's the ownership of those lines. So that goes around kind of the same question of IRBs and informed consent forms, in which it's very important for patients and donors to understand clearly who will own those lines. Will they be losing ownership of them? Will be allowed to move them to a different institution if that's what they desire? So that comes, and sometimes when the conversation is happening, it's too late. The documents have been signed and have been filed away, and there's nothing really you can do other than take your sample away from the program, because it's always you always have the right to take your sample out of a protocol. But the changes of that, sometimes you cannot do anything about it. So there's a little bit of confusion. And again, I think we'll keep talking about IRBs and informed consent forms, which are very important to understand. And if you have an opportunity to influence it, then by all means, try to do so.
Yeah.
Thank you both. All right. So what are some of the strengths of different types of cell models for different categories of rare disease?
I'm happy to start on this one. So there is different levels of utilization of the models. So typically monolayer cell lines are much easier to grow than more complex, organ on a chip or organoid models, which require a little bit more time and effort and money to be able to be successful. So the monolayer cell lines are very powerful tools for a lot of those experiments to get a project started. You can very easily modify them. You can very easily amplify them, and you can use them for high throughput therapeutic screening. We were talking earlier about doing some therapeutic screens to find new compounds. Monolayer cell lines are the place to start for sure right now. However, it is good to validate results in more complex models, and ideally in a physiologically relevant model like a mouse model, where you have an intact immune system and a stromal environment that represents what you see in the human disease. So ideally, you would have a complement of models to be able to answer your question from the very beginning to the very end. And so for every rare disease, ideally you have a cell line, you have an organoid, and you have a mouse model. However, that may be difficult to achieve.
I think the only thing that we had to add to that is, even on the monolayer models, you have sometimes it's worth going with one thing or another, right? So is it worth going all the way to an iPSC, depending on, you need to differentiate this iPSC into the tissue of your disease, because that's the only place that you will see the effect of a drug, right? Or if you can just get away with a fibroblast and that will give you the results that you need. Or sometimes you just, it's so basic that you can go away with an LCL and that's necessary, and that's, you know, for the drugs and for the disease that you're testing. I guess it's specific to the disease as well.
Great, thank you. And to follow up on that, at what stage in research should you invest in developing a cell model?
I think you can go and start, like, again, going back to the simplicity of creating a basic cell line right away. I think that at the moment that you are diagnosed with one of these disorders, contacting somebody, preferably somebody that you're comfortable with, either a researcher or a biobank, such as the one that I manage, and give blood. That's really all you have to do. So I think that's where you start. I think after that, then you when you have more results and you have a better idea of what is the next step, then you can start raising money to create more complex models, such as organoids, and even trying to get an animal model.
Yeah. I told yesterday the story of the Caring for Carcinoid Foundation, who have been involved with for many years, and they spend countless years and projects trying to generate models that were relevant for this disease, and they still really haven't found an ideal set of models. So the sooner you invest in the creation of these models for a specific disease, the more likely you are to actually be able to expand the research with those models once you have them. And it can really take some time, and it can be really difficult sometimes to do it. And other times it can be very easy. So it's worth investing on the models up front.
Perfect. Thank you. And then what are some key reasons that cell models might fail then?
I think it's what we were saying yesterday. They are living organisms, right? They are they're cells, and sometimes they just don't grow. Sometimes it's inherent to the disease itself, and a lot of chromosomal abnormalities that can create a problem, especially when we're trying to make iPSCs in the reprogramming process. Some diseases are not easy to reprogram. Some cell lines are very resistant, and we have a little bit of a trouble on differentiating them to make the stem cells. So I think a lot of times it really comes, it's natural that these things don't grow.
Yeah.
I think that we had another story for mice, right?
Yeah, I was going to talk about failure to translate.
Mm-hmm.
So when you have a really good model and you've invested a lot of time characterizing it, and you identify a really cool clinical compound that has a phenotype in a preclinical study that is beneficial, and you try to translate that to a human clinical trial, there's been examples of this translation working, and there's been examples where it didn't work because the preclinical trial in the mouse model was so different than what actually happened in the humans. So in one example, in pancreatic cancer, a trial was done for a couple of weeks in mice, had a benefit, and then they did a clinical trial in patient for months. And initially there was a benefit, but over months, the clinical outcome ended up being worse for the patients on the drug. And in fact, if they went back to the mouse and did a longer term treatment, they would have found that. So it's important to also evaluate failures to translate and to think about the translation aspect of the models, even if the models are good, because you really have to mimic what is going to be translated to the patient accurately in the model, be it a cell line or a mouse model. Otherwise, you may not be able to recapitulate the data the beneficial results that you would like to see.
Thank you. All right, so how do you see cell models addressing complex rare diseases where variants in one gene might be the causal mechanism, but there are several different types of variants?
I think we mentioned the gene editing. I think that's the biggest tool that you have right now, where you're able to take patient-derived cell models and then just play around with the different mutations and make sure that you better get a better understanding of what the disease is, what is the real cause of it, and what is the background. And I think the isogenic controls are something as well that is very useful in this matter, when you take away that gene of that variant of significance that it is, and then you see what else is behind. Also, I guess all the easy access to whole exome, genome, and whole sequencing in general, that would allow you to better understand all these little variants that we were not aware previously.
Yeah, it's been the CRISPR-Cas9 technology. I can't support enough what was just said. It's been tremendous for the research, especially for rare diseases where it's driven by a single mutation, because you can transform, very easily now cells and very cleanly. The other advantage of the current models that we have is we can generate them from patients pretty reliably, like I said earlier. So indeed, if you have even a small patient cohort and you can generate just a handful of models, if they all have the same mutation, it's great to have this biological diversity of 3 to 4 models, even with the same mutation, as long as they come from different patients. Because then you can perform the therapeutic experiments with enough biological diversity that you feel pretty confident that if you find something that works in the cells, it's going to work in every patient with that mutation. So it's great to have multiple biological replicates, is what we call them in research.
Yeah, and actually, we didn't talk about this yesterday, but it's something that is very dear to my heart. It's something as well that we keep pushing for, is the diverse of the patients under the same disease. The background, it's so important and it's not understood and is so relevant. So we need to increase the level of diversity even within the same disease. The geographically, ethnic background is so, I think it's going to be the next step of understanding so many things. And that's a way to do it. It's just creating a diversity. We talked about sex yesterday, but I don't like calling it race, but just to put a name to it, the ethnic diversity of samples is very important as well. Because that's a lot of the things in the background that are not directly related to the gene.
Yeah, thank you.
Absolutely.
And then if you guys have any questions at all for those who are attendees right now, please feel free to leave them in the chat box, and we'll be more than happy to answer those. But moving on to our next question, what kinds of skills and expertise should advocates or researchers look for when funding or collaborating with the researcher?
I think that, at least in my personal experience, communication has been key. So you need to find a partner or a collaborator or a researcher that is open about what's going on. You need to require and request and need clear results and clear answers to your questions. I think the open communication between researchers, you have to be able to see, the informed consent and make sure that it matches your needs. You need to be able to ask about what are the quality control parameters that are going to be followed in the lab. Because I see a lot of people, and I see this with people working with iPSCs, and they will come to me and be like, so-and-so will make an iPSC for $4,000, $3,000. What's the difference? Why is some people charging more or less? And as I always said, did they give you the list of the QC? What are you going to get from it? How many vials are you going to get of this type of vials? What is the QC that you're going to do on that level? So those kind of informations are very important. So you want to work with somebody that is able to provide all that and understand that you understand the disease just as well, and that you need to understand what's going on. Because I see that time and time again with patient advocates that come to me later and said, I didn't know they were going to do that. They told me they were going to create this and they didn't, or they only have one vial now. And what am I going to do now? And they won't give it to me, or they won't share. So that's the other very important thing. Do you want your samples to be shared by the community? Do you have any interest on the majority of scientists being able to access your samples? So those are the kind of things that you need to clarify before you start working with somebody.
Thank you.
Yeah. I would say on the research side, find somebody that is able to dedicate time to your question, because researchers invariably run out of time more than they run out of resources. I would say time is probably our most precious resource. So if you find a research partner that really is invested in answering the research question that you're posing with your model, then that's probably going to be a successful and productive collaboration. And on the advocate side, I would say, make sure that the expectations are in line with what's feasible, because otherwise, if the expectations are too high and it's impossible to accomplish the goal, initially, it's going to be really hard to continue building upon the initial collaboration. So you want to start small and kind of build up to a large research program. And you can do that with one research partner, but ideally you kind of build up into a larger research program through a foundation with multiple research partners. So you can have different goals and different projects that are difficult to accomplish in one single lab. So maybe not starting too fast, too big right away is my advice for the advocates.
That's great advice. Thank you. So what kinds of controls should be considered when developing or testing cell models?
We go back to that isogenic control, right? I think that's the key. That's gold standard now. When you create an induced pluripotent stem cell, you want to have your isogenic control. But I think we can forget about non-affected family members. With trios are very interesting for researchers. So we sometimes work with unaffected siblings, with or without the mutation. I think both of them are interesting for researchers. And of course, same age control, same sex control, and something what I just mentioned, the background diversity controls. Those are also interesting.
Yeah. Yeah, yesterday we had I had a discussion with a research partner, and they were looking for a normal control for an appendix cancer project, and the normal control that they had identified would come from resection of appendix from teenagers, because they're often getting their appendix taken out. But it was problematic because appendix cancer happens in 60 plus years old. So you can't really, it wasn't reasonable to try to get a normal control from teenagers, because it would have nothing to do with the biology of the 60-year-old organ. So you really need to find controls that are appropriate for what you're trying to study, which is the age control. And then the sex control is very important too. And the URM control is very important.
For you, Herve, so with cancer patients, did you guys do a lot of control on healthy tissue compared to tumor tissue?
Yeah, so we try to do as much normal controls as possible, and the organoid has made that easier because now we can grow the normal epithelium, but it also brings fresh issues with that. So if we talk about tissue, which is the easiest place to have a normal control, with pancreatic cancer, for example, the cancer that we're studying is a ductal cancer. And in the normal organ, the normal healthy ducts are a minority of the cells in the normal organ, in the normal healthy organ. So it's really hard to use the normal pancreas as a control, because your actual control cell is a minority of the population. So you have to kind of dissect the normal pancreas to get the real normal control. And in the organoid models, for example, where we can grow normal organoids from healthy patients and tumor organoids from tumor patients, those are great controls for genetic experiment and therapeutic experiments and everything else. But the normal controls, the normal organoids that we grow in the laboratory, they're proliferating. So they're actually, even though they're normal genetically, they're more representative of a wound healing phenotype, because they're constantly proliferating. Whereas the normal cells in a patient, in a healthy patient, should be homeostatic. They shouldn't be proliferating. So we try to incorporate as much normal as possible in our experiment, normal controls as possible. But we also are, very cognizant of the sort of the caveats of the culture conditions, for example, and what that causes.
Thanks.
And then I kind of wanted to go back to something you mentioned earlier, Deborah, about the importance of including a diverse background of patients. And I was wondering if you have any examples of how we can include more diverse backgrounds of patients within.
That is a current work of many researchers. It's limited because of there's a lot of ethical issues behind going and say, we're not supposed to say, but I'm going to look for African American patients, or I'm going to look for Asian patients. But we do go to the communities. We have been working with communities in a way that it's a little bit So people are doing this in New York or in California, where it's a little bit easier when we ask them for the four grandparents of the individual to be from a specific geographical location. Okay. So those controls seems to be working great. We right now just accepted a group of cell lines from a researcher that was doing a research on the Indian population, and the samples were taken in the US, but every person can guarantee that 4 of their grandparents were born in India. Okay. So that's a way to do this kind of thing. So we're trying to increase the diversity of our samples. We also have, and I think we at some point when we will talk about what is, getting an advisory committee, a scientific advisory committee. We have one, of course, and we have many members of this community that are working very hardly on diversity of samples, and that is a goal for most of the repositories now, because to be honest, the percentage of diversity is very low. So I think it's a goal for many repositories to create these models. And again, we're trying to do what I'm saying to begin with, trying to work with grandparents. But hopefully, some people are working, this is a big consortium of people working in Africa, and they're trying to increase the way that the biobanks are working. The NHGRI has now the Pan-Genome project, which takes out of, I don't know if you guys know about the 1,000 genomes, which was one of the first groups of cell lines, and they were established from diverse populations, and they're going back and trying to increase the diversity of their samples. So the Pan-Genome is working as well on that. So there's a lot of initiatives going on at the moment, trying to increase that diversity on the creation of cell lines, and then on the research itself, because I think we have that way of influencing the way the research is done.
Yeah.
Thank you.
I can add a little bit to that by just talking about the importance of just looking at the local communities around you, and integrating places that are normally not integrated into research. So, for example, when I started doing a lot of the organoid research, we were on Long Island, right outside of New York City, and we're getting a lot of the samples from New York City themselves. So the big hospitals in New York City. I'm not going to name names, but I'm sure you know who I'm talking about. And the diversity of the patient population that we're getting from there was very small. And we had to then start broadening our pool of hospitals that would submit samples to us from the surrounding neighborhoods. So outside of the, Manhattan itself. And once we did that, the diversity shut up, because there is a lot of hospitals that serve communities that are, different than the communities in Manhattan itself. That took a lot of effort. And we had one PI that was very dedicated to this, and she spent many years trying to get the other hospitals that were serving underrepresented minorities to be able to get to the point where they could send us samples. And that actually meant the creation of a whole IRB sort of department for the collection of samples, because at that time they had an IRB, but they weren't doing any research. So their IRB didn't have any research experience and how to craft a consent form for research. So it took many years just of administrative effort that was not compensated, just to be able to start getting samples into the pool. And now, many years afterwards, we have a much more diverse cohort, and we started publishing actually on some of the results that were on some of the data where we're actually seeing differences between the different ethnical backgrounds. So there's a lot of upfront investment, which is usually very difficult to do because you need to basically find the time and sponsor it yourself.
Gotcha. Great, thank you. That was very great to hear. All right. So going on, how can we ensure licenses are not hindering access to cell models by all stakeholders, including other academic groups and researchers in the industry?
I think the answer to that is change the regulations now, because we can't do that. It's important to understand what these licenses are, what kind of research is going to be being performed with the models. If you're in most of the cases, so we work with licenses specifically for the reprogramming of the iPSCs, right? So as long as you're doing research, these licenses are not, you don't have to pay any royalties for using them. If you are a commercial entity and you are going to create a product, then you need to have a license. So I guess, it really depends what the final use of your lines are. Then you have other cases. Right now we have finally the Broad Institute has gotten the rights for the CRISPR-Cas9 technology. So that actually has become a bigger deal. Of a lot of labs were using this technology while they were still arguing who had the right over the patent. And I think that now that it's established, the Broad Institute is actually coming down a little bit stronger on laboratories. So just inform yourselves. Your researchers should understand the final use of your cell lines, if it's going to be distributed, if it's going to be just shared, if it's going to be used in research, or if it's going to go and use in a commercial product, to see at what level you fall under the category for the licenses.
Yeah, just read the consent form very carefully, because all of that should be, or at least what the cell line and the model are going to be used for should be very clearly listed in there, and in relationship to commercial entities. So typically there's going to be a limitation on the commercialization of those models.
Yeah. Even like if you go to somebody like if you go to somebody like ourselves, for example, there is something called the materials transfer agreement, which is what we use to share our cell lines to researchers. Those should also be available if you're working with somebody else who is going to be distributing your cell lines. Ours is on our website. You are able to open it and read it in detail, so that in that document you'll see exactly what researchers are agreeing with each other, so us with the researchers, and then if they have to share somebody else. So all those things are detailed in there as well.
Yeah, and a lot of us follow guidance from the NIH, from the consent forms and the MTA forms, and those are available also on the NIH website.
Yeah.
I think they call it the UBMTA or something like that, a universal MTA, and we pretty much use a version of that too.
Yeah, our MTA, even though it's specific for our cell lines, is actually drafted and approved by the lawyers at the NIH.
Yeah.
So yeah, it's that's why also it's impossible to change, by the way. So if you don't agree with it, then just have to go with somebody else, because we are not going back to the lawyers at the NIH for any changes. That took years to just finalize the one that we have right now. So we're not changing them anytime soon. So even that, you have to be aware that even if you don't agree with something, then you have to perhaps go with somebody else.
That actually has become, just to follow up, that has become very common these days. There's a lot of collaborators when I ask for things where the standard response is absolutely, but the terms are non-negotiable. We're not going back to whoever, designed the terms.
Yeah.
Which, has not been an issue as long as, I've been in research, but it is definitely something to keep in mind.
I think the commercial entities are probably the ones that come up more with, oh, I like to change that or change this. But at the same time, which is good for the patients and for the advocates to know that at the end of the day, my MTA doesn't even have to do anything if you didn't consent for it. So I think at the end of the day, even if my NTA said, yes, you can commercialize this all day long, if you're signing an informed consent that says, do not commercialize my product, there's nothing I can do. So that's it. It all falls down to the consent that you have given as a patient, as a donor.
Gotcha. Yeah. And I think that kind of goes along with some ways that we can ensure making sure that patients are comfortable with working with this all is the consent. Are there any other ways that we can ensure that cell lines are used in a way to make patients comfortable other than just the consent?
They follow the consent. Find somebody that you feel comfortable working with. At least I think you have to find a researcher that, like Herve said, first of all, somebody that is really motivated and working on your particular question, somebody that is willing to share with you what's going on. I think that's very important. And then just read every single word on those consents.
Yeah. When I started in research, one of the it's still really famous now, but I think one of the worries was that patients wouldn't be aware that you're actually working on their tissues. And there's famous stories like Henrietta Lacks, where there was ethical issues with the use of the material, which were real and had to be corrected. But I find that in the last, I don't know, several years at least, it's the opposite. It's people really want you to use the resources and the samples they're providing, and they want to see research and they want to see papers and they want to see outcomes from all that material. So I feel like these days it's more about taking full advantage of what's available and not wasting it. That's the main concern. And yeah, it goes back to having the resources, which is time and money, and the space that you need to do the research, and having the people that are technically knowledgeable to be able to take advantage of the samples.
I have two thoughts on that. One, something that I was thinking about the researchers, it is my personal experience and opinion that it's better to share your cell lines. I know that as scientists, we have big egos, and a lot of scientists just feel like they've done the work, so they deserve the credit, or they want to keep going on their own little research because they put so much work on it. I recommend that you work with researchers that are willing to share information, share resources. Be in a place where you understand that the more people that have access to your samples, the better your chances of a treatment is. It's just one lab does, the work done by 10 labs is better than the work done by just one lab, even if it's the best lab. So that's the one thing. And the other thing is, you mentioned the shift, right, on what people wanted and what people want now. So our personal experience, we had our informed consent didn't have a choice for sharing genetic data because people didn't want to. People thought that it was important to keep their identity enclosed, and they didn't want people to know what kind of genetic data they have. And then one year we decided to add an opt-in of, would you allow us to share your genetic data? I can tell you that in the last 5 years probably that we added that, 99% of our donors want their data to be shared. So I think that's completely different. People think that we want to hide people, the IRBs, and they really want us to be very secretive about the data. And I keep telling you, the patients don't want that. The patients want the data to be out there. They don't care. I have a patient who keeps telling me, put my name on my sample. I don't care. Like, give them my address. Like, for the God, love of God, and who cares if they know who I am? Or is the famous Dr. Church, who has a picture. He has his sample is in a repository, and he has a picture somewhere on the internet with his GM number on his forehead, that he wrote his code for the repository and put a picture out there because that's how much he wanted people to know that his sample was available. So I think it's shifted a little bit.
Yeah. We had one of the very first patients that supported our work with the organoids when I was on the East Coast, de-identified herself at a meeting in front of 800 people, and talked about the work that was ongoing with her model and what that meant to her, what that meant to other patients. And yeah, I find that like we just talked about a lot, the desire now is to be as open with the research and the data as possible. And there clearly is a lack of, there's a disconnect between the IRB for sure, because it is lagging in terms of what it's trying to do at this time. It's more of an older thinking that they're using right now in the IRBs. So that goes to what we discussed earlier. The patients have a lot of power because they're the ones signing, they're the ones demanding that the tissue be used for research. So they can use that through advocacy groups, through foundations, to kind of, make progress at the institutional level.
Great, that's great insight. So we got about 5 minutes left, everyone. So if you have any questions at all, please feel free to put it in the chat box so we can answer them. We have time for probably about 1 or 2 more, so please feel free to put those in the chat box. And then moving along, so how can patients or researchers be stewards in ensuring research and data are not siloed and are made as broadly available as possible to academic and industry researchers? So I think you guys are all kind of talking about this with sharing the data and whatnot. Are there any other ways that we can help make sure that it's not siloed?
No, I mean, oftentimes the journals will insist that the data be published be available publicly. So like the genomic and sequencing data be available publicly, and that's great. They could also the journals themselves could do a better job, enforcing their own policies, because even though you have to agree to that and you're published in that journal for many years, the data may still not be available. So there's still improvements to be made. It's hard for the patient to really be pushing at that point. So I think it's more of a researchers and journals. But the patient should insist that if they sign a consent form for their data to be distributed, that actually happens.
Okay. That's good. All right. So next up, what is some advice you would like to give someone who finds the research process daunting?
I think my advice yesterday, and it continues to be, is talk to other advocates. You guys are like the experts on this. Other advocates have gone through the same process. They understand, they know the people, they can recommend, they can tell you. The amount of people that come to me saying, so-and-so say that I should call you. So-and-so told me that this was not the way to do this, what should I do? If you find other people with your disease, join foundations, join groups, and if there are none out there, create one. I think that's support is the best way that you can navigate these very complicated roads.
Thank you.
Yeah, it's a great answer.
That's great advice. All right. Is there anything else, any type of advice or anything off the top of your mind that you would like to let them know about on this topic before we head out?
I just want to say, by the way, at the end, so that if you guys have any questions for either of you or myself, I'm sure our information is on the agenda. If you have any questions about, what was the work that Herve is doing in California, or if you're interested in donating to the repository or finding out how we work, please feel free to contact us. It doesn't have to be through here. Maybe you need some time to think about the questions that you might have, and we'll be more than happy to answer them by email at any time.
Yeah, absolutely.
Well, thank you so much, Deborah and Herve. This was very, very informative and helpful. And thank you to all of you who attended today. Once again, if you have any questions for them, their information is on the website. So please feel free to reach out to them. Next up, we do have another session taking place from 2:30 to 3:30. So if you would like to head over to the auditorium and then click on that center image, the agenda will pop up. Go ahead and scroll down to that next session and click join. So thank you all so much again, and enjoy the rest of the event. Bye.
Thank you. Bye-bye. Bye.
Bye.